Ophiocordyceps unilateralis, the zombie ant fungus. Imagine for a moment that you're walking down the street, living your normal life with its everyday worries, when suddenly your body stops obeying you. Your legs move on their own, forcing you to march toward a destination you didn't choose, a specific place where you will meet your end. You can't scream. You can't stop. You are a passenger trapped inside your own biological vehicle. This isn't just the premise of The Last of Us, it's the daily reality in tropical rainforests for carpenter ants under the yoke of Ophiocordyceps unilateralis. This fungus isn't a simple predator, it's a molecular puppeteer of terrifying precision. The process begins invisibly. A microscopic spore lands on the ant's exoskeleton. Unlike other fungi that wait for a wound to enter, Ophiocordyceps uses specialized enzymes to dissolve the ant's armor and penetrate its body. Once inside, it doesn't kill the host immediately. That would be wasteful. Instead, it grows as a network of internal filaments, consuming the ant's non-vital tissues while leaving the central nervous system and essential organs intact. The ant keeps working, keeps feeding, completely unaware of the monster gestating inside it. But the true horror begins when the fungus is ready to reproduce. It needs very specific humidity and temperature conditions to release its spores, conditions not found on the forest floor where the ant lives. So the fungus takes control. It releases a chemical cocktail of alkaloids and neuromodulators directly into the ant's brain. Suddenly, the ant breaks away from its colony. It stops following pheromone trails and begins to climb. What happens next is what scientists call the death bite. The fungus manipulates the ant's mandibles, forcing them to clamp with unnatural strength onto the main vein of a leaf, exactly about 25 centimeters above the ground, oriented toward the north. The precision is chilling. Once the ant is anchored, the fungus kills it by devouring its internal organs and uses the corpse as a launch platform. Days later, a grotesque stalk sprouts from the back of the ant's head, a death antenna that rains spores onto its former sisters walking below, restarting the cycle. It's an evolutionary efficiency that borders on the demonic.
Toxoplasma gondii. How cats manipulate humans. If Ophiocordyceps is brutal body horror, Toxoplasma gondii is subtle psychological thriller, the spy that lives in your house and sleeps in your bed. This protozoan parasite has a single goal in life: to reach the intestines of a cat. It's the only place where it can reproduce sexually. But it often gets stuck in an intermediate host, like a rat or mouse. This is where evolution has engineered a mind control mechanism that defies all survival logic. Rodents, by instinct burned into their DNA over millions of years, fear the smell of cat urine. It's a signal of imminent death that triggers an immediate flight response. However, when a rat is infected with Toxoplasma, something fascinating and disturbing happens. The parasite hijacks the rat's amygdala, the part of the brain responsible for processing fear. It doesn't just eliminate the terror of the cat's scent, it rewires neural circuits to turn that fear into sexual attraction. The infected rat doesn't merely stop fleeing, it actively seeks out the cat. It's drawn to the predator that will devour it. The parasite, in essence, serves its host up on a silver platter to complete its own life cycle. The rat dies, the cat feeds, and Toxoplasma wins. But here's where the story becomes unsettling for us. Humans can also be hosts for Toxoplasma. It's estimated that a third of the world's population carries this parasite in their brain, latent. For years, it was thought to be harmless in healthy adults, but recent studies suggest darker correlations. Infected individuals have been observed to have slower reaction times and a higher likelihood of being involved in traffic accidents. Even more disturbing is the manipulation hypothesis, which suggests links to schizophrenia, bipolar disorder, and increased risk-taking and aggression. Is it possible that a sudden urge to drive too fast or a lack of fear in a dangerous situation isn't bravery at all, but a microscopic parasite clumsily trying to lead us toward a predator that no longer exists in our urban environment? The line between the self and the tenant blurs dangerously.
The hairworm that forces crickets to drown. Let's talk about biological claustrophobia. Imagine having inside you a being that grows to be 3 or 4 times longer than your own body, coiled and compressed among your organs, waiting for the moment to escape. This is the fate of crickets and grasshoppers infected by Spinochordotes tellini, also known as the hairworm or horsehair worm. The larvae of this worm are microscopic and live in water, where they're ingested by insect larvae that later become mosquitoes, which are then eaten by crickets. Once inside the terrestrial cricket, the worm begins to grow, and it grows enormously. It absorbs nutrients directly from the host's body, turning the cricket into a mere living container. But the hairworm is an aquatic animal. It needs water to reproduce and die. The problem is that the cricket is a land creature that normally avoids large bodies of water to avoid drowning. This is where chemical mind control comes into play. When the worm has reached maturity and occupies almost the entire body cavity of the cricket, it begins secreting proteins that mimic the insect's neurotransmitters. These chemical signals sabotage the cricket's navigation system. The insect starts behaving erratically and, in a perversion of its natural instincts, desperately seeks water. Upon finding a pond or pool, the cricket throws itself into suicidal immersion. The moment the host touches water, the horror scene begins. The worm, sensing the liquid, starts exiting the cricket's body, piercing its exoskeleton. It's a nightmare vision, a long, writhing filament emerging from a much smaller insect, like a living hose unspooling endlessly. The cricket is left as an empty, drowned shell, while the worm swims free to find a mate and continue the cycle of aquatic terror.
The parasite that castrates and changes the sex of crabs, Sacculina carcini. If there were an award for the cruelest and most transformative parasite, Sacculina carcini would take gold without competition. This parasitic barnacle doesn't settle for controlling behavior. It rewrites the fundamental biology and identity of its victim. Its target is the green crab, and its method is total anatomical violation. Sacculina begins life as a microscopic larva searching for a vulnerable crab. When it finds one, it doesn't merely attach, it injects itself through a joint in the crab's armor, shedding its own shell and becoming an amorphous mass of cells that invades the crustacean's interior. Once inside, it extends root-like tendrils throughout the crab's body, slowly digesting it from within and absorbing nutrients. But the true monstrosity lies in what it does to the reproductive system. Sacculina castrates the crab, atrophying its gonads and rendering it sterile. The crab will no longer waste energy reproducing. All its energy will go to the parasite. Then Sacculina grows an external brood sac on the underside of the crab, exactly where females would carry their own eggs. And here comes the final twist: maternal mind control. If the host is female, the parasite chemically tricks her into caring for that sac of parasites as if they were her own offspring. She will clean it, oxygenate it, and protect it. But if the host is male, Sacculina alters his hormonal balance to feminize him. The male crab begins to behave like a female. His claws stop growing for combat, and his abdomen widens. He too will care for and protect the parasite's eggs with a mother's devotion, even performing spawning movements to help release Saccharina larvae into the water. The crab doesn't just lose the ability to reproduce, it loses its sex and its purpose, turned into an eternal nanny for the species that destroyed it.
The jewel wasp that walks cockroaches like dogs. In the parasite world, there are butchers and there are neurosurgeons. The jewel wasp, Ampulex compressa, is without a doubt an elite neurosurgeon. Its goal isn't to kill its prey, the American cockroach, but to subdue it and keep it fresh. The cockroach is much larger and stronger than the wasp, so a direct attack would be suicidal. The wasp needs to eliminate the cockroach's will, not its life. The attack is a precise choreography of 2 stings. The first is a quick strike to the cockroach's thorax, temporarily paralyzing its front legs. This gives the wasp just enough time to prepare the master stroke. The second sting is a macabre work of art. The wasp inserts its stinger directly into the cockroach's brain. It doesn't inject lethal venom, but a very specific neurotransmitter blocker, such as octopinine, that regulates motivation and complex movement. The result isn't total paralysis. The cockroach can still walk, still feel, but it has completely lost the will to do anything on its own. It becomes a docile zombie. The wasp then bites off one of the cockroach's antennae and drinks a little hemolymph as a snack before pulling the antenna like a dog leash. The cockroach, capable of defending itself but chemically stripped of its survival instinct, follows meekly into the wasp's burrow. Once inside, the wasp lays an egg on the cockroach and seals the entrance. When the larva hatches, the cockroach is still alive and docile, waiting patiently in the dark. The larva chews a hole into the cockroach's side and enters. It feeds on the internal organs in a specific order, leaving the nervous system and heart for last, ensuring the meat stays fresh and alive as long as possible. The cockroach is eaten alive from the inside, silently and without resistance, a victim of nature's most perfect chemical lobotomy. Leucochloridium, the snails with psychedelic eyes. If nature had a psychedelic horror genre, Leucochloridium paradoxum would be its star. This parasitic flatworm has a life cycle that requires moving from a snail to a bird. But snails are shy, live in the shadows, and hide from birds. The parasite needs to change that, and it does so by turning the snail into a living neon sign. The snail ingests the parasite's eggs through bird droppings. Inside the snail, the larvae develop and migrate to the snail's eye stalks. This is where the most grotesque physical transformation on this list occurs. The worms invade the eye stalks and swell, becoming pulsating sacs of bright colors, greens, yellows, and oranges with concentric rings. They don't stop at changing color. The worms begin to pulse rhythmically, contracting and expanding rapidly. The visual effect is identical to a tasty caterpillar or grub wriggling around. The snail, now effectively blind, with eyes turned into hypnotic fishing lures, also undergoes a behavioral change. The parasite manipulates its brain to abandon the safety of darkness and shadows, forcing it to climb to the highest parts of plants in broad daylight, where it's perfectly visible. To a hungry bird, those pulsating eyes look like juicy caterpillars. The bird swoops down and pecks off the snail's eyes to eat them. Amazingly, the snail often survives this mutilation, and its eyes can regrow, only to be infected again. Meanwhile, the parasite reproduces inside the bird's intestine, completing its cycle. It's a light show designed to lure death from the sky.
Rabies, the virus that makes you fear water to spread. We often think of parasites as worms or insects, But viruses can be the most insidious manipulators, and none is as legendary or as terrifying as rabies. Rabies isn't just a disease, it's a biological demonic possession that has inspired werewolf and vampire myths for millennia. Its mind control mechanism is brutally simple and devastatingly effective. It hijacks the limbic system to maximize its own transmission. The rabies virus is transmitted through saliva and enters the nervous system, slowly climbing toward the brain. Once there, it causes massive inflammation, encephalitis, but in a very specific way. It alters the host's behavior to make it hyperreactive and aggressive. The virus needs the animal to bite another to jump to a new host. It turns a docile pet or a rational human into a machine of irrational fury, stripping away inhibitions and amplifying the fight response. But the most cruel manipulation is hydrophobia. The virus concentrates in high amounts in the salivary glands. To ensure the saliva stays virus-laden and ready to infect, the pathogen must prevent the host from swallowing it. How does it do this? By causing painful, uncontrollable spasms in the throat whenever the host tries to drink water. Over time, even the sight or sound of water triggers absolute panic. Driven mad by thirst, yet physically unable to drink, the host begins to drool profusely, creating the characteristic foam at the mouth. That foam is concentrated virus, ready to be injected. Rabies doesn't just kill you, it turns you into a biological weapon, using your own thirst and aggression as vehicles for its immortality, torturing you to the very last second of consciousness.
Could this happen to humans? Evolutionary theories. After this parade of horrors, the inevitable and chilling question arises, are we humans, with our higher consciousness and technology, safe from this kind of biological control? The short and terrifying answer is probably not. In fact, it may already be happening in ways we're only beginning to understand. From an evolutionary perspective, the human brain is just another biological substrate, complex but manipulable. We've already seen how Toxoplasma can subtly alter our personality and behavior. But there's an emerging field of study suggesting that our oldest tenants might be pulling the strings, our gut microbiome. We have trillions of bacteria in our intestines, outnumbering our own human cells. These bacteria aren't passive passengers. They produce neurotransmitters like serotonin and dopamine, the same chemicals that regulate our mood, anxiety, and happiness. Recent studies suggest the microbiome can influence our food cravings, making us desire sugar or fat to feed specific bacterial strains. More unsettling still, imbalances in this flora have been linked to depression and autism. Are our cravings really ours, or are they chemical signals sent by a bacterial colony demanding to be fed? Moreover, the history of viruses is written into our own DNA. Approximately 8% of the human genome consists of remnants of ancient viruses that infected our ancestors millions of years ago. Some of these viral genes are essential to our survival, such as those that allow the development of the placenta. We've domesticated some monsters, yes, but what if a new pathogen, perhaps released by melting permafrost or emerging from a zoonotic mutation, evolved not to kill us, but to change us? In nature, the leap from controlling an ant to controlling a complex mammal is large, but not impossible. Rabies already crosses that boundary. If a parasite developed the ability to press the right keys in our prefrontal cortex, manipulating our beliefs, our fears, or our tribal loyalties to favor its transmission, would we notice? Or would we think, like the ant climbing the leaf, or the rat seeking the cat, that the idea, that impulse, was entirely our own? The true nightmare isn't losing control of our body, it's losing the certainty that our mind belongs to us.